Hello and welcome to my presentation on antibiotic resistance in cattle. This is an important topic because antibiotic resistance poses a significant challenge to public health globally. In the context of cattle, the misuse and overuse of antibiotics in livestock farming not only jeopardize animal health, but also increase the risk of resistant bacteria transferring from animals to humans. So let's talk a little bit about the basic cellular structure and function of microorganisms. There are two different categories of cells. Human and animal cells fall into the eukaryotic category, and bacteria fall into the prokaryotic category. The difference between these cells lie within their structure. Human and animal cells, or eukaryotic cells, have a nucleus that's enclosed by a nuclear membrane, and that is where all their DNA is stored. Bacterial cells, or prokaryotic cells, do not have a nucleus. Their DNA floats freely in the cell cytoplasm. Bacterial cells are also much smaller and simpler than human or animal cells. They lack many of the organelles that are found in eukaryotic cells, like the mitochondria and the endoplasmic reticulum. Bacteria have a cell wall made of peptidoglycan, a unique substance that provides structural support and protection. Human and animal cells do not have a cell wall. They only have a cell membrane that is more flexible. Human and animal digestive tracts contain trillions of bacteria that help digest food, produce vitamins, and protect against pathogenic bacteria. Bacteria also live on the skin and other body surfaces where they can help protect against disease-causing organisms and contribute to the immune system's proper function. While many bacteria are beneficial or harmless, some can cause diseases if they invade parts of the body where they're not usually found, or if the immune system is compromised. DNA is the blueprint for all cellular functions, and in bacteria, it's typically a single circular chromosome located in the area called the nucleoid. Unlike the multiple chromosomes in the eukaryotic cells, bacterial DNA is compact and often includes additional small circles of DNA known as plasmids. Plasmids can carry genes for traits like antibiotic resistance, and they are key in horizontal gene transfer between bacteria. Bacteria reproduce asexually through binary fission, a process where the bacterial chromosome replicates and the cell divides into two genetically identical daughter cells. This allows for rapid population growth. During replication, mutations can occur, potentially leading to traits like antibiotic resistance. Antibiotics typically kill successful bacteria, but those with mutations that have resistance can survive and multiply. Besides mutations, bacteria can acquire resistance through horizontal gene transfer. They can exchange genetic material, including plasmids carrying resistance genes, with other bacteria. This exchange can occur even between different species of bacteria, and it's a major factor in the rapid spread of antibiotic resistance across bacterial populations. Pathogenic bacteria can cause diseases in animals and humans by invading the body and evading the immune system. They can enter through ingestion, inhalation, or breaks in the skin, and then attach to host cells using structures like pili or adhesions. Once inside, these bacteria can invade deeper tissues, even crossing barriers like the intestinal wall or the blood-brain barrier. They then evade immune detection by hiding within host cells. These pathogens can cause direct tissue damage through the production of enzymes and toxins that disrupt cell membranes and cellular functions. They can also cause indirect damage by triggering severe inflammatory responses in the host. Antibiotics are powerful drugs that combat bacterial infections by targeting specific cellular processes essential for bacterial growth and survival. There are different classes that work in different ways. There are cell wall synthesis inhibitors such as penicillins and cephalosporins, and they prevent bacteria from forming the cell wall, which maintains the cell's integrity and shape. Without a proper cell wall, bacteria cannot withstand osmotic pressure, leading to cell lysis and death. There are protein synthesis inhibitors such as tetracyclines, macrolides, and aminoglycosides, and they bind to bacterial ribosomes. This is the machinery responsible for synthesizing proteins. By interfering with ribosomes, these drugs effectively halt the production of essential proteins, crippling bacterial growth and function. DNA replication inhibitors such as fluoroquinolines target bacterial enzymes responsible for DNA replication and repair. Inhibiting these enzymes prevents the bacteria from replicating its DNA, thereby stopping cell division and multiplication. There are RNA polymerase inhibitors such as rifampin, and these bind to the bacterial RNA polymerase, an enzyme critical for synthesizing RNA. Blocking RNA polymerase stops the transcription process, preventing the bacteria from synthesizing RNA and producing proteins. Bacteria can develop resistance to antibiotics through different mechanisms driven by mutations in their DNA. These mutations can change the structure of the molecule targeted by the antibiotic, such as the ribosomal binding sites or enzymes, preventing the antibiotic from binding effectively and rendering it ineffective. Bacteria can increase the activity of efflux pumps, which expel antibiotics from the cell before they can act, or produce enzymes like beta-lactamase that degrade the antibiotic. Other mutations may decrease the permeability of the bacterial cell membrane, limiting the entry of antibiotics and reducing their effectiveness. These adaptations allow bacteria to survive and multiply even in the presence of antibiotics. A few of the short-term consequences of antibiotics include reduced effectiveness, where the bacteria develop resistance to the existing antibiotics and become less effective. So situations where common infections were easily curable with antibiotics become harder to manage and cure. It increases healthcare costs because treatments for antibiotic resistance infections typically require longer durations at the hospital. This includes more intensive care and the use of more expensive or alternative medications, and increased mortality from treatable infections. With the reduced effectiveness of antibiotics, mortality rates for infections that were previously considered manageable can increase. There are also long-term consequences to antibiotic usage. Antibiotics are heavily used in livestock for disease treatment, prevention, and growth promotion, with about 70% of medically important antibiotics in the U.S. allocated for animal use. This extensive usage poses significant risks by encouraging the development and spread of antibiotic-resistant bacteria. These bacteria can flourish within animal populations and spread to humans through the food chain, environmental runoff, and direct contact, leading to difficult-to-treat infections. The implications for human health are profound, with antibiotic-resistant infections contributing to at least 700,000 global deaths annually. These infections can often result in longer hospital stays, increased healthcare costs, and the need for more costly, less effective treatments. Economically, antibiotic resistance could cost the U.S. up to $1 trillion by 2050 if the trends persist. There are several ways we can take action to address this problem. We can start by banning the use of antibiotics as growth promoters and only using them therapeutically. We can promote good agricultural and animal husbandry practices such as better hygiene, good nutrition, and the use of vaccines, and this can reduce the need for antibiotics. We can educate farmers about the risks associated with antibiotic misuse and the importance of adopting alternative disease control strategies. We can raise consumer awareness about the benefits of choosing meat products from animals raised without unnecessary antibiotics. We can continue to enforce strict regulations and monitor antibiotic sales and usage. We can establish penalties for noncompliance and incentives for farms that adhere or go beyond regulations. And finally, we can collaborate with supermarkets and meat retailers to promote and preferably display products that comply with antibiotic stewardship standards. By implementing these strategies, local communities can play a significant role in reducing antibiotic resistance, ensuring sustainable farming practices, and promoting public health and food safety. For more information, please visit any of the sites listed below.